Yeah, thank you very much for the invitation. I'm very happy that I have the opportunity to present my research to you. So now I don't see anyone. So if anything, if there is anything, so bad connection or an urgent question, then you really need to interrupt me. But that's no problem. You can interrupt me anytime. Just that as a starting point. Yeah, the title of my talk has already been mentioned: effects of expectancy on the processing or in the processing of actions and their outcomes. And I chose the the term expectancy, although I could have also said prediction. So the title of the talk could also be effects of prediction on the processing of actions and their outcomes, because expectancy and prediction, they are of course closely related. And predictions are also a central aspect of many current theories of brain function and of cognitive function. For example, talking about sensory processing, then these theories, and the most prominent one is predictive coding theory, they propose that our brain constantly predicts the sensory input. So let's give a brief example, a very simple one. Here you see a button that you can find in local trains in the S-Bahn, for example. I'm sure many of you have seen it before or something similar. And this button here can be pressed. And when it is pressed, then, for example, it changes the color. But not only that, also when the train stops, it changes the color. And this indicates that the button is activated. And this is just an everyday example that there is certain sensory input that is predicted or expected somehow. And according to these theories that state that we constantly predict the sensory input, uh, we don't process at every point in time the whole sensory surrounding, the whole sensory environment, but what we process is the mismatch between what we predict and what actually occurs. And that is nothing else than a prediction error. And the prediction error is a central concept that we will encounter during the talk, and I'm sure you have also encountered it very frequently already. So the prediction error is a central aspect, and from a computational point of view, it is also much more efficient to process the prediction error compared to the complete sensory input at every point in time. But not only in sensation, predictions and expectations play a large role, but also in other domains or cognitive functions. Predictions have been proposed to be very important, and this is also reflected in the concept of the brain as predictive machine. For example, predictions have been also related to higher cognitive functions such as social cognition. So we predict, for example, what other people will do, how they will behave, what action they are going to perform, or how they will feel like. And also, we make predictions about our own um, emotional states. And when we think about that, predictions and expectations play a role in many different, or can play a role in many different cognitive domains, then we may ask different questions. For example, we may ask, is there a similar role of prediction across the different domains? And another question could be, are there similar or are there distinct prediction mechanisms within one domain and between domains? So is the type of prediction that we use always the same, or are there differences in the type of prediction? And these kinds of questions will also be tackled by the studies that I'm going to present. So the talk is divided into three parts. First, I will talk about predicting the sensory consequences of actions. Then I will talk about predicting cognitive consequences of actions. And the last very short part will be about predicting observed actions. Let's start with predicting the sensory consequences of actions. Whenever we perform actions, then these actions lead to sensory consequences. And in our perception, we need to be able to distinguish between the sensory consequences of our own actions and that part of the sensory input that is not caused by ourselves. So let's again make a simple example. When I move my arm around here in the air, maybe you can see that, how I do this, then this leads to a wind-like sensation on my arm and on my hand. And of course, I know that this wind-like sensation, this air pressure, is caused by my own movement. But if I was not aware of this, then the same sensation would be interpreted completely differently. Then that could mean, this wind-like sensation could mean that a door is open somewhere or a window is open somewhere. So somehow we need to distinguish between the sensory input caused by ourselves and sensory input that is not caused by ourselves. And one important phenomenon that plays a role or may play a role in this is the so-called sensory attenuation. What does that mean? Actually, in the literature, sensory attenuation has two meanings that are related. The first meaning is that there is reduced perceptual intensity for self-produced stimuli compared to external stimuli. The most famous example for this is that we are not able to tickle ourselves. The tickle sensation is only there if someone else tickles us, but if we try to do that ourselves, the sensation is not the same. And this reduced perceptual intensity for self-produced stimuli may also be the reason for why our own voice we hear usually less loud than other people hear our own voice. And the other meaning of sensory attenuation is that there is also reduced neurophysiological activity for self-produced stimuli. And these two phenomena seem to be related also, but they have been shown to be not the same. But this is only a side note. We will focus on the second part, on the reduced neurophysiological activity for self-produced stimuli. And this can be examined in the lab in different modalities. And I will give you an example that we have used also ourselves in the auditory modality, because there a very simple paradigm has been used and is still being used in the literature. This is called self-generation paradigm. The classical version has just three experimental conditions. The first one is that participants are asked to press a button in regular intervals, and this button press causes a sound. This is here the upper row in this graph on the left side. And then there is another condition with external sounds. So the same kinds of sounds are played, but they are not self-produced. They are just played like this without a preceding button press. And then there is a third condition, which is only a control condition, because in the self-produced sound conditions, of course, there is a motor action going on, and this may also cause neurophysiological activity. And in the motor control condition, buttons are pressed, but no sound is elicited, and this controls just for the motor activity or for the neural activity caused by the movement itself. And when we now examine how the sounds are processed, the self-produced sounds and the external sounds, and measure brain activity, for example, with EEG, then we see typically the following. We see that for self-produced sounds, the amplitudes of event-related potentials are reduced compared to external sounds. We see this here in two prominent event-related potential components, the N1, the first negativity, and the P2. And the green line here is for the self-produced sounds, and the blue line is for the external sounds. And you can see that the amplitude for processing the sound is reduced when the sound is self-generated. So we have reduced N1 and P2 event-related potentials for self-produced compared to external sounds. How does that come? How is that possible? And one explanation for this is the so-called forward model. What does that mean? That means whenever we perform an action, then not only a movement command is generated in our motor cortex and then goes to the spinal cord and is transferred to the muscles in order to execute the movement, but a copy of the motor command is sent to a forward model. And this forward model then predicts the sensory consequences of our upcoming action even before the action is executed. And then the predicted sensory consequences and the actual consequences are compared to one another. And when they match, then there is no prediction error, and then this sensory attenuation is occurring. And Where in the brain could be such a forward model? The cerebellum has been linked to this forward model prediction because the cerebellum, with its vast amount of neurons and the resulting computational machinery, is a perfect candidate for housing such a forward model. And the question then is, or no, before we come to the question, another slide on the cerebellum and the forward model. So not only from a theoretical point of view, it makes sense that the cerebellum houses the forward model, but there is also anatomical evidence because the cerebellum is connected with the cerebral cortex via closed loops, as you can see here in this figure. So these loops between the cerebral cortex and the cerebellum, they originate in the cerebral cortex, and then via the pons, they go to the cerebellar cortex, from there to the cerebellar deep nuclei, from there to the thalamus, and back to the cerebral cortex. And these loops, they are closed loops, and they are topographically organized. That means that they originate from the same part of the cerebral cortex where they also end. And the starting point of such loops can be in the motor cortex, but it can also be in non-motor cortices, for example, in the prefrontal cortex that is more strongly related to cognitive functions. And the first question that we are asking is there a role of the cerebellum actually really in sensory attenuation? Is the cerebellum responsible for sensory attenuation? And this question has already been asked by a group in Leipzig about 10 years ago, or more than 10 years ago. They applied the self-generation paradigm that I introduced in patients with cerebellar lesions. And here we see the event-related potentials, both for healthy controls and for patients with cerebellar lesions, both in response to self-produced sounds and to external sounds. So in the upper row, we have in each of the three figures that we see there, we have the data from both controls and patients. But in the second and third row, we have patients and controls separately. And let's focus on this because there it is easier to see. In the middle row, here we have the controls, and we have the typical pattern that the N1 and the P2 amplitude here at these three electrode sites is reduced for self-produced compared to external sounds. So self-produced sounds is the dotted line. In the patients, there is a different picture, especially here in the early processing in the N1. There is no, or at least a strongly reduced sensory attenuation or reduced effect here in the patients because the amplitudes of self-produced sounds and of external sounds, that they are much more similar than in the control participants. However, when we look at the later processing here in the P2 time window, there we see also an attenuated amplitude for self-produced sounds for the patients. So to summarize, we have a reduced N1 attenuation in cerebellar lesion patients, but intact P2 attenuation. And we can say, yes, we have evidence that the cerebellum contributes to the processing of sensory action consequences. When we think about our first example again, that there is this button in the S-Bahn and that it changes its color when we press it, then we can say that an own action, an own button press, is not the only possibility to predict the sensory input. Actually, it's much more common probably that we observe someone else pressing the button, and we can also use observed actions to predict sensory input. And the question that we ask then is, what about the prediction of sensory consequences of observed actions? Does that work similarly compared to the prediction of sensory consequences of own actions? And in this context, it is interesting that the forward model has been linked to the mirror neuron system that is involved in action observation. So when we observe someone pressing a button or performing any type of action, this is probably processed, at least in part, first in the visual system in the superior temporal sulcus that is specialized for the perception of biological motion. And from there, the mirror neuron system is then activated. So in the parietal and in the prefrontal cortex or inferior frontal cortex. From there, the primary motor cortex can be activated. And from there, the same processes can happen as if I was performing the action myself. So that means that again, the cerebellar forward model could be involved, at least in principle or in theory. And the question that we asked then, is there sensory attenuation in action observation? And for this purpose, we developed an observation version of the self-generation paradigm that I introduced. So we had two participants in the lab in parallel. One was the actor and one was the observer, and they were sitting next to each other in front of a computer screen, as we can see on this picture here. For the self group, for one group of participants, the different conditions were the same as those that I already presented. So in one condition, self-produced sounds were played, in another condition, external sounds, and there was a motor control condition. The observation group then had the corresponding conditions. So there was one condition in which they observed the actor pressing buttons, and this button press that they observed elicited a sound. Or in the motor control condition, they observed button presses that did not elicit a sound. So the task of the observation group was just to observe what the active participants did. The external sound condition was actually the same because both participants were just hearing sounds and no action was proceeding. What about the event-related potentials now? And this is what we see here. So in red, we see event-related potentials to those sounds that were caused by an action on the left side for the performers and on the right side for the observers. Then you see two other lines here, the black and the blue ones. They belong to two conditions with external sounds. So this is the only difference to the standard paradigm. We had two conditions with external sounds, one in which the external sounds were played separately and one where they were intermixed with self-produced sounds, but that doesn't matter too much. Let's just focus on the red line and how they differ from the other conditions. You can already guess here that in the N1, when we focus on the N1 first, that in the N1, there is an attenuation only in the performers, but not in the observers. But this is more clearly seen here when we look at the amplitudes as a bar graphs. Here we see that there is a reduction of the N1 amplitude, but only for the actors, not for the observers. And this was also reflected in the statistical analysis. When we look at the later processing here in the P2 time window, there is again a different pattern. When we look at the bar graphs for this, then we see that there is attenuation for the P2, both in performers and in observers. So the amplitudes here of this P2 component, they are reduced compared to the external sounds in both performers and observers. So we have something like sensory attenuation also in observation, but it just affects the later processing in the P2 time window. And the next question that we ask now is if there are similar prediction mechanisms. We see already here that the processing differs depending on whether I press the button myself to elicit a sound or observe someone, but still we see that there is some kind of prediction in both going on, in actors and observers. And it could be that for this delayed effect in observers, also cerebellar prediction mechanisms play a role. And this is what we tested in another study. And here we again assessed the processing of action-generated sounds, but this time with fMRI. And we had control conditions where either no button press was performed or no sound was played, or both. So the structure of the control conditions was a little bit different that you can see here. So we can think of the paradigm, first of all, as a 2x2 design. Participants pressed either a button that was followed by a sound or that was not followed by a sound, or they heard a tone that a sound that was not preceded by a button press, or they neither pressed a button nor heard a sound. This is the new control condition, so to speak, that we called visual only condition because participants just had to watch at a fixation cross. But then we were interested, of course, in the comparison of active button presses and action observation. So we had this 2 by 2 design again by 2 because there was another set of 4 conditions where participants could observe button presses by others on the computer screen that were again either followed by a sound or not, or there was just the sound without a button press, without an observed button press, or neither an observed button press nor a sound. And what we did was we used multivariate pattern analysis to classify, based on the activation pattern, the 8 conditions with a linear support vector machine classifier. And we did also a whole brain analysis, but in our main analysis, we used the activation pattern in predefined regions of interest that we were particularly interested in. We did this with a between subjects leave one out procedure. And we also determined the most likely class partition. So the most likely, let's say, clustering of experimental conditions based on Bayesian hypothesis testing on the confusion matrix. And we focused, or here in this talk, I would like to focus on 2 regions. First of all, on auditory cortex, because we think that something like sensory attenuation needs to take place in auditory cortex. And we asked, is there is a differential activation for sounds that are preceded by actions or by observed actions in the auditory cortex? And the second question targeted the prediction mechanisms, if there is evidence of prediction at all. And here we focused on the cerebellum. We wanted to know if there is a differential activation pattern for actions or observed actions that are or are not followed by sounds. Or in other words, does it make a difference for the cerebellum if a button press leads to a sound or doesn't lead to a sound, and if it's an active or an observed button press? Starting with the auditory cortex. So the auditory cortex was defined as primary auditory cortex plus higher auditory cortex. So it's a little bit more broadly defined. First of all, we had here in the right auditory cortex highly significant classification accuracy of 32%. 32% doesn't sound much, but remember we had 8 conditions, so chance level was 12.5%. And this explains why 32% is quite highly significant. So what was the most likely class partition? What conditions could the activation in the right auditory cortex differentiate? In the right auditory cortex, conditions with and without sound could be differentiated. This is something we would really expect from the auditory cortex, but this is like a proof of principle. So the activation seemed to reliably differ between those conditions where there was a sound and where there was no sound. That's no surprise. But the activation in the right auditory cortex could also differentiate between conditions with action-generated sounds and external sounds. So also these 2 conditions here in yellow, no matter if the sound was preceded by an own action or an observed action, could be reliably distinguished from all the other conditions. And on top of this, there was also a difference between conditions with sounds that were generated by own versus observed actions. Also, this made a difference to the activation in the auditory cortex if the sound was caused by an own or by an observed action. So all in all, we found evidence of sensory prediction. This is in line with the finding of sensory attenuation, both for action and for observation. Although I have to say that we did not test for attenuation of the signal. We just tested whether the activation pattern in the auditory cortex differs between these 8 conditions. Now let's turn to the cerebellum. What did we find there? We looked at the motor cerebellum, and within the motor cerebellum, we had several sub-regions of interest. For the left motor cerebellum, we again had highly significant classification accuracy. And also here, we want to look at which conditions could be differentiated. First of all, conditions with active responses differed in their activation pattern from the rest. So all those conditions where actively a button had to be pressed showed a different activation pattern compared to the rest. This is also something we would really expect. But what could also be distinguished was own actions with and without auditory consequences. So it did make a difference for the cerebellum if a button press was followed by a sound or not. And the same pattern was also found for another region of interest in the cerebellum, interestingly on the right side for lobule 6. That was exactly the same class partition. So this finding is in line with a cerebellar forward model prediction, but only for own actions. So we saw that there is prediction also going on for action observation, but it seems that the cerebellum is not involved in this. The cerebellum seems to be involved only in predictions for own actions. Let's summarize this. Perception requires the distinction between self-produced and external stimulation. And to achieve this, we need a neural mechanism that is able to predict sensory action consequences. Comparing the predicted and actual sensory action consequences can contribute to the ascription of agency. This is something that we didn't mention so far. So we can use these predictions also in order to determine if some stimulation in the environment was caused by ourselves or by someone else, or if we cannot ascribe an agent to this stimulation. This applies to both own and observed actions. One potential mechanism seems to be prediction based on afferents copies that are fed into a forward model. But it seems that this mechanism, at least from our data, only works for self actions and not for observed actions. So we are still searching for the prediction mechanism that is involved in action observation. So then we would come to the second part of the talk, predicting cognitive consequences of actions. And here we would like to talk about learning from positive and negative feedback in particular. Let's look at our example again. This button that I told you about in the S-Bahn, of course, we don't press the button because it changes the color. This is maybe a nice side effect, but we follow a certain goal with the button press because the button is actually there to open the door. And if our goal is to open the door and we press it, the button, and then the door actually really opens, then this is positive feedback. This shows us that we have done it correctly. Now, you might say there is nothing to do wrong about this because there is only one button, but still you could press it with different durations. You could either press it or swipe over it. And the feedback that you get tells you that yeah, your goal was reached and that you did it correctly. And in the lab, we can study feedback-based learning with different kinds of paradigms. One typical paradigm would look like this, that we give participants two stimuli they can choose from, and then they choose one of them and they get feedback for their choice, either by showing just on the screen that this was correct, or also in another way. But yeah, this would be an example for correct or positive feedback. But of course, they can also get the opposite. They can get negative feedback. Then they would show on the screen that they performed an error. And as I said, feedback can be given in different ways, either like this or with monetary reward. So for correct responses, they can get a reward, and for negative consequences or for negative feedback, they can be punished or don't get a monetary reward. It has been shown that in this kind of learning, there is a central role of the prediction error because no learning takes place when there is no prediction error. The feedback needs to be surprising, at least to some degree. If we can perfectly predict already the consequences, so positive and negative feedback, then no learning is taking place. Unexpected positive feedback increases And unexpected negative feedback decreases the likelihood that a certain behavior is shown again in the future. And this is also known as reinforcement learning. And interestingly, as you are probably aware of, this prediction error that is central for learning is reflected in the activity of single dopamine neurons in the midbrain. We know that when a monkey is given a reward, then the activation of dopamine neurons increases, the firing rate increases in response to the reward. When a conditioned stimulus predicts the reward, then we don't see an activation change in response to the reward itself when it is perfectly predicted, but we see the increase in activation already in response to the conditioned stimulus. And interestingly, when a reward is predicted but does not occur, then we see a dip in the dopaminergic activity here in the firing rate of the dopamine neurons. That means that both positive and negative prediction errors can be or are reflected in the firing pattern of dopamine neurons. And this signal is then projected to other parts of the so-called reward system. And the structures that belong to this are, for example, the nucleus accumbens or ventral striatum, but also the dorsal striatum and the medial prefrontal cortex, including the anterior cingulate cortex. In all these regions, prediction error related signals have been identified either with single cell recordings in animals or also with, for example, fMRI. But interestingly, um, we can also examine feedback processing with a different method in humans with EEG or with event-related potentials. fMRI is, of course, very important to localize, spatially localized brain activity. But when we have now seen the single cell recordings, then the temporal resolution is also very important. And EEG offers the opportunity to have a high temporal resolution, although, of course, the spatial resolution is very bad or even non existent. But we know that feedback stimuli in the event-related potential elicit a negativity around 250 milliseconds after a feedback stimulus is presented. This was first described around the same time in the mid-90s when also the dopamine neuron studies were conducted. And this negativity you can find here in the EEG signal here around, as I said, 250 milliseconds after feedback for both positive and negative feedback, but it is usually more pronounced for negative feedback, as you can see here also. What I didn't mention before, in EEG research, very often the negativity is plotted upward. That was already the case in the traces that I showed before for this N1 amplitude, for example. So don't be confused. Negativity means upward usually. And this signal has been labeled feedback related negativity or feedback error related negativity. More recently, it has been proposed that this negativity that we see is actually a different component, an N2 component that is always elicited by feedback, and that when reward is given, a positivity is superimposed on the signal. And that is the reason why for positive feedback, the signal is less negative or more positive than for negative feedback. I don't want to go into this discussion of the literature. Um, we just look at the signal here in this time window, no matter if we call it feedback related negativity or reward positivity, as it has recently been labeled. What has been shown now several times, and what we also found in our own studies, is that the amplitude of the signal in this time window reflects a prediction error. And here's one recent example from our lab. We see here how the amplitude changes with higher prediction errors, so more surprise on the right here in the axis, on the x-axis, the surprise is increasing. And the blue line is for positive feedback and the green line is for negative feedback. What we found, and also others, is that the dependence of the signal from the size of the prediction error is only there for positive feedback, not for negative feedback. But there are also other studies suggesting that it can also reflect the prediction error for negative feedback. Nevertheless, or one way or the other, it is interesting that with this signal on the scalp that we record on the scalp, we can find a neural correlate of prediction error processing. Neural sources for this signal here in this time window have been described in the striatum and very recently with intracranial EEG also in the caudal anterior cingulate cortex. And one interesting question that we addressed recently is there a role of the cerebellum also in predicting these positive and negative feedback action consequences? As I said at the very beginning, one question in these different cognitive domains is if there are prediction mechanisms similar across domains. And we have already seen that the cerebellum contributes to the processing of sensory action consequences. And now we ask whether the cerebellum does something similar for cognitive action consequences like positive and negative feedback. And this brings us back to the forward model because it has been proposed already that the forward model is not only important for predicting sensory action consequences, but also for predicting cognitive action consequences. So this figure that we see here is not exactly the same as the one that I showed before, because here when you look on the right side, then you see that the cerebellum here is made responsible for predicted cognitive consequences of actions, whatever that is. It's a very broad term. But one thing of these predicted cognitive consequences, one example could be whether an action leads to something positive or something negative, which is more cognitive than this example that we had first, whether a certain stimulus like a sound is elicited or not. And in a current or just finished DFG project that was run or headed by Jutta Peterburs, professor at the Medical School Hamburg, in cooperation with the University Clinic Essen, with Dagmar Timmann-Braun, and the University Clinic Düsseldorf with Martina Minnerob, they looked exactly at this question. And actually, two experiments on the role of the cerebellum on feedback learning and feedback processing were conducted, one with patients with cerebellar stroke and one with cerebellar single pulse TMS. But I would like to focus on the first study on the patients with cerebellar stroke. We see here for patients and control, so the patients with cerebellar stroke, how they processed feedback. And the figure that we see here is a little bit different compared to the ones that we saw before. That's why I briefly explained this to you. So we don't see negative and positive feedback processing here in one figure, but they are separate. In the upper line, there is negative feedback processing, and in the lower line, there is positive feedback processing. So that is one important difference. Then on the left side, we see the controls, and on the right side, we see the patients. And you can still see that there are two lines in each figure. What are these two lines? The two lines, they differentiate between feedback with a high prediction error and feedback with a low prediction error. So feedback that is more or less surprising. And what you can already guess from this graph here is that in the controls, it seemed to make a difference whether feedback was more or less surprising, because the lines, at least here in this gray shaded time window, they diverge. Whereas for the patients, it didn't seem to make a difference. So they didn't seem to process feedback differently depending on the size of the prediction error. And this was actually also reflected in the statistical analysis. Here on the left side, we see how the amplitude of the feedback related negativity depended on the prediction error separately for negative and positive feedback. And here again, you can see that in the controls, the prediction error is reflected, but somehow a little bit different compared to what I've shown you before, because here the effect is stronger for negative feedback. Nevertheless, in the patients, we don't see that the feedback processing depends on the prediction error. So we have altered prediction error coding in the patients with cerebellar lesions, and this was further examined with voxel-based lesion symptom mapping to show where or which parts of the cerebellum or in which part of the cerebellum gray matter density was linked to aberrant prediction error processing. And we found correlates of this in cruise 1, cruise 2, lobule 7b, and 8a. So in these parts that are also colored here, gray matter density was related to the absence of prediction error processing in cerebellar lesion patients. Importantly, we have to stress that no learning deficits were found in the patients. So they processed feedback differently, but they could still learn from feedback. And it's an open question how this works if they used compensation mechanisms. But it's not unusual to find something like this for cerebellar lesion patients in the literature. So we showed now that the cerebellum does not only contribute to the processing of sensory action consequences, but also cognitive action consequences like positive and negative feedback. Now, when we think back about the sensory action consequences, there we asked, what about action observation? And we did the same, or the same question can be asked for positive and negative feedback learning. And thinking about our example, again, when we want to know what we have to do in order to open the door of the S-Bahn, we don't have to press the button ourselves. We can watch someone else to learn from the feedback this other person receives. Actually, it can be highly advantageous to not learn from own actions all the time, but first to look what others do and see and let them get negative feedback, for example, in order to avoid negative feedback for ourselves in the future. And in the lab, learning by observation can look like this. So participants don't press a button themselves or don't choose a stimulus themselves. They observe the choice of someone else and observe the feedback that the other person gets. And then we test whether they learn from this observation. And I don't want to go too much into detail, but we have conducted many studies on the comparison between active and observational learning, and we found that the processes seem to differ. For example, the feedback-related negativity, this ERP component in the EEG, reflects a prediction error for active but not for observational learning. And with fMRI studies, we also found evidence that some brain regions are more strongly involved in prediction error coding for own actions compared to action observation, including the dorsal striatum. But also we found initial evidence that the medial cerebellum is more strongly involved for action than for action observation. So we have evidence that also here in learning from positive and negative feedback, the cerebellum is involved, but only or at least more strongly for actions, self actions compared to action observation. The last study in this part, before I come to the final part, asks now if there are other factors that might affect feedback processing. You remember at the beginning we asked, is there only one prediction mechanism or maybe several prediction mechanisms? We already know there must be more than one because the cerebellum is sometimes more and sometimes less involved. But there are still other factors that might affect the prediction and in this sense also feedback processing. And here I would like to present very recent data from a study that was just accepted for publication last week. And this was somehow related to the problem of credit assignment. What is that? Let's look at a simple example from an experiment in our lab again that we have already seen. So participants choose here this left stimulus, and then they get reward for their choice. Even in this very simple and lab example where we try to exclude all other aspects, the situation may not be as clear as it seems because participants may ask themselves, what exactly do I get the reward for now? Do I get it for performing a certain action, so for pressing a button with my left hand, or get I the reward for the stimulus that I've chosen, or maybe even a combination of both. For example, do I get reward only when this stimulus that is highlighted here is shown on the left side, but not when it is shown on the right side, or when I choose it with the left button press and not with the right button press? And in everyday life, this problem of credit assignment, so the problem, what I what is it that I'm rewarded for, is much more complex, because when you experience something positive or negative, then this can be related to something you just did before, but it can also be related to something you did some minutes or even hours or days or months before. And this is, of course, then very important to learn when we want to learn from feedback, that we know for which action or stimulus we get reward. And we had one assumption that was based on previous research. We assumed that during feedback processing, a representation of the event that is associated with the feedback is reactivated. So if this is the case, then feedback processing would depend on the associated event, and the binding of feedback and event could be mediated by the hippocampus. It has been shown that the hippocampus can be involved in feedback processing. So if I refer feedback to a stimulus, for example, or to an action, this should be somehow reflected in the feedback processing itself. That was our main assumption. And more concretely, we thought that the ventral visual pathway that is important for visual processing is involved in feedback processing when the feedback refers to a visual stimulus that was shown before. For example, when participants choose a visual stimulus. And how did we look at this? We looked at another event-related potential component, the N170, because the N170 has been related to visual processing, especially to the processing of faces, but also other visual stimuli. And the experiment that we conducted was the following. There were two conditions, and every participant took part in both conditions. And the first one looks very familiar because it is similar to what we have already seen. Participants are presented with two stimuli on the screen. They choose one of them, and then they get positive or negative feedback, here indicated with this red cross for negative feedback, or this green sign for positive feedback. But in another condition, participants did not choose between two visual stimuli, but between two auditory stimuli. So they wore headphones, and on the left and on the right ear, they heard different melodies. And with a button press, they could choose now between the left or the right melody, and received feedback for this choice. So only in the visual condition, the feedback referred to a visual stimulus. In the auditory condition, feedback referred to an auditory stimulus. And we did this with immediate and delayed feedback. This is also a factor that may affect feedback processing and that we studied extensively, but I don't want to go into detail concerning this. And this was a within subjects design, as I already said. And first of all, we found that participants could learn the associations in these studies. So they learned equally well to choose between visual stimuli and to choose between auditory stimuli. And we saw a pronounced N170 component in all the different experimental conditions. So the N170, as I said, is a different event-related potential component. At first sight, it looks similar as the feedback-related negativity, but it's a different component because we see that we have the ERPs here at lateral electrode sites, P7 and P8. And for example, the feedback-related negativity would be not pronounced here at these lateral electrode sites. And here we see the topography. And here you can again see that this component is most strongly pronounced at the lateral electrode sites. So it's really something different. And it has not been so much related to feedback processing before. And in this context, it is our first interesting finding was that the N170 component that, as I said previously, was more related to visual processing and not to feedback processing, that actually this component also reflects a prediction error for negative and positive feedback. As you can see here in this graph, for different conditions, we see here only at electrode P8 the result pattern for auditory stimuli. So when participants chose between auditory stimuli or visual stimuli, or whether feedback was delayed or immediate. And in all these conditions, the N170 amplitude is modulated by the size of the prediction error. And this again shows um, how important predictions and prediction errors are, because they seem to affect processing across a variety of different domains. Now, coming to our research question, this prediction error processing in the N170 was particularly pronounced for visual stimuli and for delayed feedback and over the right hemisphere. As I said, we only see data from electrode P8, so all this is over the right hemisphere. And what you can guess from the lines here is that the prediction error effect, reflecting how strong the slope of these lines is particularly pronounced for especially for positive feedback again, for the choice between visual stimuli and for delayed feedback. So it did make a difference whether the feedback that participants received referred to auditory stimuli or to visual stimuli. Just for completeness, of course, we looked also at the feedback-related negativity. We also found effects of the stimulus modality there, but we did not find an effect on prediction error processing, depending on whether the choice was between auditory or visual stimuli. Let's summarize this part before I come to the final part, which is only one slide. So predictions and expectations play a critical role in learning from positive and negative feedback. And the prediction error is reflected in scalp-recorded ERPs, event-related potentials in humans, not only in the feedback-related negativity, but also, as we saw, in the N170 component. The cerebellum appears to contribute to feedback processing, but again, only to active and not to observational feedback processing. The mechanisms of feedback prediction and feedback processing depend on a number of different variables, for example, on agency. So whether I choose myself or observe someone, as I said, then it seems to depend on the event that is associated with the stimulus. We saw this example that it makes a difference if the feedback refers to an auditory or a visual stimulus. And the mechanisms may also depend on feedback delay. This is also, as I said, something we study quite a lot. There are also differences depending on whether the feedback is given after 1 second or, for example, after 7 seconds. Okay, as I said, I would like to very, very briefly also talk about the very last part, because it about predicting observed actions, because it somehow rounds up the whole thing, at least in my view. Because we said at the beginning, our brain is thought to make predictions all the time. And our everyday life is not structured like an experimental trial. So it's not always there is an action and then there is a consequence, and action and consequence. But the action itself, when we observe an action, this can also be is also a visual stimulus, right? So this visual stimulus can also be more or less expected and more or less predicted. And for example, about 20 years ago, an event-related potential component has been described that is more pronounced for observed errors than for observed correct responses. That was interesting because for own actions, there is such a component already longer known, the so-called error-related negativity. And then it was interesting to see that when I observe someone perform a wrong action, for example, pressing a wrong button, that this elicits a larger negativity than when I observe someone performing a correct response. But there was one important confound, because in many situations, when we observe an error, this error is more unexpected than a correct response. And that was also the case in this example here from one of our own studies. So maybe what is processed by the brain is not whether an observed action is an error or correct, but maybe it's just the expectancy that drives this processing. And what we did, we thought about a study in which errors can be expected or unexpected. And I don't want to explain in detail how we did this. I can do this later if you are interested. It was a false belief task, so there was also an aspect of perspective taking involved. But when we did this, actually, we found a larger negativity for observed correct responses. And this was strong evidence that what the brain processes is really more or less the unexpectancy of the observed action. And an observed action is then not so much different compared to negative or positive feedback, which can be more or less expected or unexpected. When I meet someone or see a person, then I make predictions about what this person is going to do next. And when the person does something unexpected, then it is just processed differently compared to when this person does something expected. And there might thus also be a prediction error for observed actions. And in a newer study, this effect is already 10 years old, we found that this effect is also modulated by trait empathy, which involves also perspective taking. Just this as a roundup. And then I would like to briefly summarize overall predictions and expectations modulate brain responses to sensory and cognitive action outcomes. And across domains, there can be a similar prediction mechanism, for example, involving the cerebellum. And this is in line with the idea that there is a domain general role of the cerebellum in the sense of Schmarmann's universal cerebellar transform. However, across and within domains, there may be different prediction mechanisms. And for example, for action observation, we have not really found the prediction mechanism yet. Predictions and expectations also affect, as we saw on the last slide, the processing of observed actions themselves. And this may be a link to higher cognitive functions like social cognition. And with this, I would like to end. I would like to thank all the people that are still or were in my lab that contributed to these studies. I would like to thank Jutta Peterbus very much because, as I said, she headed the project on the role of the cerebellum in feedback processing in cooperation with Dagmar Timmann-Braun and Martina Minnerop, whom I would also like to thank very much for the cooperation. And I would like to thank the DFG for funding parts of this research and you for your attention.